Welcome everyone. Today I will talk about the use of diffusion imaging, diffusion microstructure, and diffusion tractometry in the study of brain aging. I'll specifically focus on the major trends and themes that have emerged in the use of diffusion imaging in the study of brain aging, and I'll end the presentation with some current limitations and what I think are future applications of diffusion to add to our current understanding. I want to emphasize here that this is a very active area of research and that this lecture cannot comprehensively describe the great number of published studies. Instead, I will try to provide a decent introductory background as a starting point for follow-up investigation. Here you can see a histogram showing the number of publications in a PubMed search of diffusion MRI and the terms aging or Alzheimer's disease. Of course, this is an overestimate. Not all of these studies have directly examined brain aging or Alzheimer's disease using diffusion imaging, but the trends are correct. Essentially, you could see that there's an explosion of publications in recent years, with the publications in 2021 reaching into the hundreds. We'll not be talking about all of these studies, however, we'll try to highlight some of the major themes of this work. Although the methods have greatly advanced over the years, it might not be surprising to know that the study of brain aging with diffusion began shortly after the initial descriptions of the diffusion MRI method, and publications date back at least 30 years now. Here's one of the earliest, if not the earliest, study in the literature to focus specifically on aging, published in the Journal of Magnetic Resonance Imaging in 1994 by Gideon and colleagues. The main finding of the study was that there was a significant increase in the apparent diffusion coefficient with increasing age in what was called the normal appearing white matter, the non-lesioned white matter. The authors concluded their paper by speculating on some of the possible mechanisms of these changes, namely changes in myelin-associated bound water, demyelination, Wallerian degeneration related to cell loss, and enlarged Virchow-Robin spaces. By this time, a good deal was already known about the histopathologic changes in the brain with typical aging and due to age-associated disorders such as Alzheimer's disease and cerebrovascular disease. However, then, as now, it was not particularly clear how to define aging and what the boundary between age and disease is. The introductory paragraph of this review from Mrak and colleagues demonstrates the sentiment at the time. Aging, along with taxes, is one of the absolute certainties of life. Everyone knows what aging is and can recognize it when they see it. It is thus truly amazing how little we understand about the process. Although many diseases become more likely as one ages, stroke, Alzheimer's disease, Parkinson's disease, it's not at all clear that any diseases are inevitable consequences of aging. Indeed, it is difficult to provide even a non-controversial, non-trivial definition of aging. A typical definition might be a progressive unfavorable loss of adaptation resulting in decreasing expectation of life with passage of time. That is, the older you get, the closer you are to death. The age-associated changes that they highlighted here included gross changes in brain weight, which were most attributable to changes in white matter, and particularly frontal white matter, increases in ventricular volume, decreases in neuronal cell size, decrease in the number of myelinated fibers, and reduction in dendritic complexity, as well as changes in neurochemical expression, lipofuscin or neuronal inclusions. And they also noted that there's an increase in the rate of changes after about 60 years of age. Here are some of the major questions and themes in the study of aging using diffusion imaging that we'll touch on here. This includes examination of temporal models of age trajectories, for example, whether the effects of age show an acceleration as noted in prior pathology studies, determination of whether there are regional patterns of tissue vulnerability, including voxel-based, region of interest, and tract or attractive interest-based vulnerability, sex differences in diffusion properties, and sex by age interactions, studies exploring the health and lifestyle correlates and mechanisms of age-related change in microstructure, and differentiating typical aging from the effects of age-related disease. Before we begin, however, it's important to note that, as with any other study, results in the interpretation of diffusion studies of brain aging will be dependent on the specifics of the participant sample examined. As noted in the previous slide, there really is no uniform process of aging. A range of health conditions are typical with advancing age. Some are more typical, some are more rare. Some are relatively benign, whereas others are more severe. For example, hypertension is relatively common in older adults, and elevated blood pressure has impacts on the nervous system without major symptoms in the absence of cardiovascular or cerebrovascular event. Alzheimer's disease is a bit less prevalent, but is severe in the symptomatic phase. Additionally, pathologies from conditions such as Alzheimer's disease impact the nervous system years or decades prior to prominent symptoms and a clinical diagnosis. Aging could therefore be considered a composite of the various influences on the nervous system given the health conditions that are common for a given age range in a selected cohort. It's important to be aware that we are likely examining the impact of several things in composite. It may therefore be challenging to attribute effects to any one mechanism or develop a universal theory of how the brain ages. It's also crucial to consider the study design when comparing studies. A significant majority of the studies in the literature use cross-sectional analysis to infer the effects of age, and I'll be talking about many studies using cross-sectional design. It's very likely that there are cohort effects that contribute to the age effects measured cross-sectionally. For example, dietary differences in the 1940s compared to the 1990s may lead to group differences that are not based in change over time, but instead due to premorbid group differences. A selection bias may also be found in sampling the healthier and more intact older adults that are able to participate in significantly taxing imaging research studies and did not have the more severe negative health consequences of aging. Finally, it's important to consider what portion of the age span the study data come from, as effects may differ in different segments of the adult age span. A major advancement in diffusion imaging technology was the advent of methods for quantification of anisotropic water diffusion, and a resurgence of studies were conducted to use these new methods in the study of brain aging. Several early studies are listed at the bottom of this slide. These data are from publication Neurobiology of Aging in 1995. These are voxel-based maps demonstrating group differences in fractional anisotropy in older adults 60 to 80 years of age compared to younger adults 20 to 40 years. And the bottom panel shows middle-aged adults aged 40 to 60 years compared to the same younger adults 20 to 40 years. The color scale shows the strongest statistical effects in yellow. As you can see, the effects are prominent in the frontal white matter and extend posteriorly to parietal white matter and periventricularly. Effects were similar in the mid-aged adults, but the effect was a bit reduced in magnitude. On the right, you can see a diffusion tensor map overlaid on the effect map. Based on the anatomical information, prominent effects were in the inferior orbitofrontal white matter, including projection regions of the forceps minor, the uncinate fasciculus, and the inferior frontal occipital fasciculus. The take-home message from these early studies include fractional anisotropy as a sensitive indicator of age-associated processes. These processes can be quantified as early as mid-age and may increase in severity in later aging, and that there's some selectivity to the regional vulnerability, with frontal white matter being particularly affected. Several studies examine the spatial patterns of age-associated changes in tissue integrity measured by diffusion imaging to determine whether any basic principles of neural vulnerability could be determined. A few of the hypothesized models or patterns described are demonstrated here. For example, the top image is from Pfefferbaum and colleagues in 2005 and shows the difference between younger and older adults in fractional anisotropy stepping through the coronal plane of the brain from the anterior slices on the left to the posterior slices on the right. The effect in p-value is demonstrated on the bottom of the plot. As can be seen by this plot, the younger adults have significantly greater fractional anisotropy in the anterior slices of the brain, and this effect is greatly reduced about midway through the brain. A similar anterior to posterior gradient was described by Lebel and colleagues using tractography to examine fiber tracts within the corpus callosum, with the orbitofrontal and anterior frontal fibers being most vulnerable to the effects of age. Given the prominent vulnerability of frontal white matter, others propose the last in, first out theory of aging brain vulnerability, in which late developing white matter tracts, such as those comprising frontal white matter, may be particularly vulnerable to advanced age. Similarly, theories posit that regions of the brain late to develop evolutionarily may be particularly vulnerable to the effects of age. Although it is the case that certain late developing regions such as frontal white matter are vulnerable, direct tests of the hypothesis that there's a pattern related to developmental maturity patterns has not borne out. This figure is from work by Yeatman and colleagues examining this question directly. The left panel shows a series of hypothetical piecewise linear models that are consistent with the last in, first out hypothesis. In each hypothetical track lifespan curve, the age that the MRI parameter reaches its mature value is negatively correlated with the age it begins to decline. The right two panels show the age of maturity plotted against the age of decline as determined from the piecewise linear models, with the effect related to diffusion data in the diffusivity plot all the way on the right. These data are not consistent with the last in, first out hypothesis, as there is no systematic relationship between the age at which the tract matures and the age at which it begins to decline. In addition to these findings of a lack of vulnerability based on developmental trajectories, it should also be noted that although strong effects can be measured throughout much of the frontal white matter, these effects are not uniform. These data show results from a focused analysis of prefrontal white matter using diffusion fractional anisotropy, demonstrating that, as mentioned previously, effects in inferior regions such as medial and lateral orbitofrontal white matter may be most prominent. As can also be seen here, white matter bordering on the ventricles shows a strong age effect. However, other portions of the frontal white matter are less significantly affected. The image below shows a relative effect size analysis demonstrating the regions of the white matter show the greatest statistical differentiation from effects in other regions. As can be seen here, relative effects are greatest in regions of frontal white matter. However, the effects are also greatest in regions bordering the lateral ventricles. Additionally, regions bordering the posterior lateral ventricles show greater effects than several more anterior regions, demonstrating again that it's not a simple anterior to posterior gradient. However, these data may suggest that there's an anterior to posterior gradient in the effect of age in certain regions such as the periventricular white matter as well as the callosal white matter. I've already noted some of the tracts that have been found to be most vulnerable to the effects of age. I do want to highlight this recent study by Cox and colleagues using tractometry in data from 3,513 participants from the UK Biobank to examine the effects of age in various microstructural parameters. In the right panel, you can see the effects of age, laterality, sex, and age by sex interactions. Effects of age were most prominent in anterior and posterior thalamic radiations and association fibers, including the inferior frontal occipital fasciculus and uncinate fasciculus. These results may suggest that there is some tract-selective vulnerability to the effect of age. These data are also in alignment with the voxel-based studies demonstrating prominent effects in frontal white matter and white matter that borders the ventricular systems. Although there were sex differences in microstructural measures, age by sex interactions were minimal and inconsistent, suggesting limited differences in the effect of age on tract microstructure between men and women. This presentation is focused primarily on white matter microstructure measured by fractional anisotropy and mean diffusivity, as these measures were the focus of several early studies. These studies generally demonstrate regional decreases in fractional anisotropy and increases in mean diffusivity with age. Of course, in the subsequent years, several additional microstructural modeling procedures were advanced that all may provide unique information regarding microstructural alterations in the brain. However, it's important to note that these microstructural measures tend to be highly correlated and may not provide much in the way of unique information. For example, the top image here shows a correlation matrix of several microstructural measures, including the mean axial and radial diffusivities, fractional anisotropy, and higher order measures, including the mean axial and radial kurtosis. The heat color scale shows the amount of overlapping variance from 0 in red to 100% in bright yellow. Overall, all measures are strongly correlated, with greater than 40% of the variance overlapping for almost all pairs, and most being over 70%. Highest correlations can be found between the radial and mean diffusivity, the radial diffusivity and the fractional anisotropy, and the mean and radial kurtosis. The most unique pair of metrics is the radial kurtosis and the axial diffusivity, but they still share 36% of their variance. Thus, these data demonstrate considerations when interpreting any unique microstructural effects. Although sharing a significant amount of their variance, there is still some degree of unique information provided by each measure, and each measure may be somewhat differentially, regionally sensitive to the effects of age. This color map corresponds to the microstructural measure that showed the greatest age effect for that region. For example, these light blue frontal regions showed the greatest effect in radial diffusivity, whereas the more anterior regions showed greatest effects in fractional anisotropy. These data generally demonstrate the need to better understand unique microstructural aspects of brain aging and what this information might provide regarding specific mechanisms of age-associated brain changes. As noted earlier, there are characteristic trajectories of development throughout the white matter described previously. Age of peak development is typically determined by the point at which diffusion microstructural measures reach their peak, for example in the case of fractional anisotropy, or trough, for example in the case of mean diffusivity. The image on the left from Lebel and colleagues demonstrates the age of peak fractional anisotropy on top and age of minimum mean diffusivity on bottom for various fiber tracts. This paper nicely defined the age trajectories across various fiber bundles. The simple point that I would like to make here is that there's a wide range in developmental peaks across these fiber systems extending into the mid-40s. We must therefore be careful to account for these continued developmental maturation effects when attempting to model and understand the senescence effects of aging. The images on the right are from a more recent study by Beck and colleagues. They performed a combined cross-sectional and longitudinal analysis of aging trajectories in the white matter. Some global measures are demonstrated in the top panel with the fractional isotropy, mean diffusivity, and axial diffusivity values by age on the x-axis, and longitudinal points connected by lines. The peak or dip can be seen in these data around the age of 40 or a bit later for the axial diffusivity. On the bottom, you can see the idealized curves for a range of microstructural measures with advancing age. These data demonstrate the need to account for dynamic patterns of white matter changes across the adult age span. As noted previously, these trajectories are fairly similar across microstructural measures, although each measure has some variation in their age trajectory. Additionally, the authors noted that traditional diffusion tensor metrics, such as fractional isotropy, was among the most sensitive to the detection of age-related variation among the several that were tested. Several studies have examined associations between age-associated changes in diffusion microstructure and cognition. Generally, these studies show significant associations between diffusion microstructural measures and cognition across multiple cognitive domains, with substantial effects in fluid cognition and executive function. Much of these associations are mediated by age, and removal of age variance from the models weakens the associations. Microstructural integrity does not seem to have a specific effect on functional networks. That is, although there may be associations with white matter integrity and functional connectivity measures, these associations do not seem to be specific to the structural connections of those functional networks, at least within the typical range of variation. On the right is one relatively recent study by Berger and colleagues demonstrating associations between executive function and diffusion metrics. These are diffusion metrics on the left within white matter lesions, and on the right, diffusion metrics and normal appearing white matter. There are therefore associations between age and variation in white matter microstructure and cognition. However, the degree to which these associations are anatomically or cognitively specific is still unknown. I will just briefly note that, of course, there are a number of conditions that increase the prevalence in older adults that can contribute to the effects detected in the analysis of brain aging. Alzheimer's disease and cerebrovascular risk and disease are two of the most prevalent age-associated conditions that affect the brain. On the left, you can see the effect of Alzheimer's disease compared to otherwise matched older adults. As is clear, Alzheimer's disease results in regional reductions in tissue integrity, and differential regional effects can be detected with different microstructural measures. Hypertension is common in older adults. However, hypertension is a somewhat arbitrary categorical variable based on blood pressure exceeding a certain level. The top right image demonstrates that there's a quantitative association between blood pressure and diffusion microstructure throughout much of the white matter that extends to blood pressure variation in the typical non-hypertensive range. These results demonstrate the complex influence of typical health and physiology on brain white matter. Individuals with more significant cerebrovascular dysfunction often have white matter lesions, also known as white matter hyperintensities, or white matter signal abnormalities. These white matter lesions are often considered indicators of microvascular disease that are independent from the health of the normal appearing white matter where lesions are not found. The bottom right image shows that in fact, white matter lesions are an indicator of the health of the normal appearing white matter as well. This image shows the statistical association between white matter lesions and diffusion anisotropy, showing strong associations with greater lesion burden by being associated with reduced white matter integrity throughout the normal appearing white matter. These data show just a few of the various health-related influences that can impact the results in the study of brain aging. There are several limitations in the use of diffusion imaging for the study of aging that must be considered when evaluating this research. I've listed some of them here and broke them down into methodological and conceptual categories. On the methodological side, diffusion images are distorted and have artifacts and bias. How does this affect the microstructural quantification? For example, some of the regions showing greatest age-associated microstructural effects, such as orbitofrontal white matter, have greatest susceptibility artifacts. It is possible that this contributes to the effects measured with age. Diffusion images are relatively low resolution, and the brain atrophies with aging. There is therefore potential bias in the partial volume contamination, which refers to the mixing of tissue constituents within a voxel, with the older adults having more gray matter or CSF contributing to some regions measured, which would represent a macrostructural as opposed to a pure microstructural effect. This effect is particularly pronounced in the fornix, which is a region that has been highlighted in various diffusion studies. Increased motion with aging. Prior studies have noted increases in scanner motion with aging and age-associated disease, and this motion affects microstructural quantification. It's therefore important to consider methods to correct for motion in statistical models and other aspects of image processing. Microstructural measures are affected by multiple processes, so any effect may be some aggregate that's not an indicator of any specific process. As discussed previously, it will be challenging to use diffusion imaging to elucidate one specific process or pattern of aging. Diffusion tractography can be biased in brains with damaged tissue. For example, failure to meet fractional anisotropy thresholds for tractography due to fractional anisotropy reductions with age and disease has resulted in the erroneous conclusion that the entire tract is completely degenerated. However, the result is simply due to a technical limitation in the tractography procedures. Diffusion microstructure has relatively low test-retest reliability compared to other modalities such as structural MRI, and longitudinal effects may be small. These factors make longitudinal measurement or measurements of intervention effects challenging. Conceptually, given the use of diffusion imaging in tract-based anatomy, it's often assumed that effects will occur at the anatomic level of the tract. In other words, that there are specific tracts that are uniformly vulnerable. However, voxel-based analyses do not support tract specificity in the effects of age, or that this might be a rare effect. That is, regions showing effects on voxel-based maps do not form tracts, but instead seem to follow other aspects of the neuroanatomy and physiology. Thus, analysis at the tract level can be counterproductive. When tracts are specifically vulnerable, it's possible that damage will be restricted to a specific portion of the tract. This could be examined using along-the-tract-based measures such as the measures available in Tracula. This image shows a study of patients with Alzheimer's disease using a path of interest procedure that measured from the anterior to posterior portions of the hippocampal cingulum bundle. Effects with Alzheimer's disease were most significant in the anterior portions of the fiber tract in the left and right hemispheres, including the tracts bordering the regions that accumulate early primary Alzheimer's disease neuropathology, such as the entorhinal cortex. The effects somewhat tapered off when moving more posteriorly. These findings may be consistent with Wallerian degeneration of cortical regions due to neurodegeneration in tract target regions. This is just an example to demonstrate the utility of these types of procedures in the interpretation of diffusion results. Here's another issue that's often not considered that was described in a recent publication by Figley and colleagues. This issue pertains to the fact that cerebral white matter voxels contain multiple fiber populations with different trajectories, which renders the microstructural measures highly sensitive to the relative volume fractions of the various fiber populations, the microstructural integrity of each constituent of the fiber population, and the interaction between these factors. Given these complex biophysical underpinnings, conventional interpretations can lead to incorrect conclusions. The image in the center of the slide demonstrates the primary fiber orientations on the top, the secondary fiber orientations in the middle, and the tertiary fiber orientations on the bottom. As can be seen, there are regional patterns where the secondary and tertiary fibers are more prominent. Given these patterns, the image on the right demonstrates a couple of hypothetical scenarios. The top shows a scenario in which one of the crossing fiber populations is altered, the horizontal fibers here, and the other remains intact. In the second case, both fiber populations are damaged. In the top case, the fractional anisotropy is actually increased as opposed to decreased because loss of the crossing fiber population results in greater restriction of diffusion along vertical fibers. In the bottom scenario, although both fiber populations are damaged, there is still no overall change in the fractional anisotropy because there is no difference in the uniformity of diffusion restriction when both fiber populations are damaged. This additional consideration demonstrates the care required when interpreting any diffusion imaging result. I've alluded to some of the concepts that require further attention throughout this presentation. For example, what is the mechanistic basics of age-associated changes in diffusion microstructure? What mechanisms facilitate spatial patterns of vulnerability? Can microstructural measures provide specific indicators of histologic or pathologic processes? Are microstructural changes biologically meaningful? How can we optimize longitudinal quantification of microstructural parameters? Can microstructural measures be used to determine the impact of therapeutic interventions? This image on the right is from a recent review of associations between modifiable risk factors and white matter integrity measured by diffusion imaging. The take-home message from the review is that there are several modifiable factors that could be treated to prevent or slow age-related white matter decline. To date, there have not been large randomized studies to examine this robustly. However, smaller observational interventional studies have produced promising results. Thus, the use of diffusion to measure treatment efficacy will be an important future direction. Given the versatility of diffusion imaging, the multiple microstructural measures possible, the anatomical definition provided through diffusion modeling, diffusion imaging will continue to have a significant role in the study of brain aging and age-associated diseases. Thank you and enjoy the conference.